In today's video, we're going to go through everything you need to understand about binary fission, which is the method by which prokaryotic organisms such as bacteria divide and reproduce. We will first explain how binary fission functions, and then we will move on to understanding how to calculate how fast bacterial populations can multiply under the right conditions. Before we dive in, it's important to make one thing absolutely clear: binary fission is not the same process as mitosis or meiosis. Mitosis and meiosis occur in eukaryotic cells, which are more complex and include cells found in plants, animals, and fungi. Binary fission, on the other hand, occurs exclusively in prokaryotic cells, such as bacteria, which are far simpler in structure. If we take a moment to look at the structure of a single bacterial cell, we can observe that it typically contains a cell wall, a cell membrane, cytoplasm, a large circular strand of deoxyribonucleic acid, which holds all the crucial genetic information, and several small loops of deoxyribonucleic acid, known as plasmids. These plasmids often contain non-essential genes that may offer certain advantages in specific environments. Additionally, some bacteria may also have a flagellum, which is a tail-like structure used for movement, though not all bacteria have one. Binary fission is a process in which one bacterial cell divides into two new cells, and because bacteria are unicellular organisms, this form of cell division is also a form of reproduction. This is known as asexual reproduction, meaning that the bacteria do not require another organism to reproduce. Instead, one cell replicates and splits, forming two genetically identical cells. Now let's take a closer look at how binary fission actually occurs. Before the bacterial cell can divide, it must first increase in size so that it can support two new cells. It must then replicate its genetic material, meaning the large circular deoxyribonucleic acid strand must be copied, and so must all of the smaller plasmids. After this replication step, the two large circular deoxyribonucleic acid molecules move to opposite ends of the cell, ensuring that each daughter cell will receive one copy. The plasmids, on the other hand, are distributed somewhat randomly, meaning one daughter cell might end up with more plasmids than the other. Once this is done, the cell begins to grow a new cell wall in the center, and when that wall is fully formed, it splits the original cell into two separate and genetically identical daughter cells. Just like that, one bacterium becomes two. What makes this process particularly significant is how fast it can happen. Under ideal conditions, some bacteria are capable of dividing every 20 minutes. Since each division results in the population doubling, the growth is exponential. For example, one bacterium becomes two, two become four, four become eight, and so on. This exponential growth means bacterial populations can increase extremely rapidly. If the conditions remain ideal and a single bacterium keeps dividing every 20 minutes, then over the course of 5 hours the population could exceed 250,000 bacteria. Now, let's learn how to calculate this sort of population growth. Suppose we are told that a bacterial cell has a mean division time of 30 minutes, and we are asked how many cells it would produce after 3 hours. First, we need to convert 3 hours into minutes, which is 180 minutes. We then divide this total time by the division time of 30 minutes, giving us 6 division cycles. Since each cycle doubles the number of cells, we start with one original cell and multiply by 2 6 times. That is 2 to the power of 6, which equals 64. So, after 3 hours, there would be 64 bacteria. Let's do another example. A petri dish contains 1,000 bacterial cells, and each cell divides once every 20 minutes. How many bacterial cells will be present after 4 hours? First, we convert 4 hours into minutes, which is 240 minutes. We then divide that by 20 minutes, the mean division time, to get 12 division cycles. Then, since we are starting with 1,000 cells and each cell doubles 12 times, we calculate 1,000 multiplied by 2 to the power of 12. 2 to the power of 12 is 4,096, and when multiplied by 1,000 it gives us a total of 4,096,000 bacterial cells after 4 hours. It's important to remember that how quickly bacteria divide depends on both their species and the conditions in which they are kept. For optimal growth, bacteria need warmth, moisture, and a rich supply of nutrients. If any of these are missing or less than ideal, then the bacteria will divide much more slowly, or in some cases, not divide at all. So, to summarize, binary fission is a process that allows prokaryotic organisms like bacteria to reproduce quickly by duplicating their genetic material and dividing into two new identical cells. Under ideal conditions, this can lead to extremely fast population growth, which is why bacteria can spread so rapidly. Understanding how to calculate the growth of bacterial populations using mean division time and the total time is a useful skill, especially in exams and in real-life applications such as healthcare and microbiology.